This lecture will cover part 1 of chapter 17, the specific and adaptable defenses of the host. In particular, today we'll cover the overall working of the immune system, and we will look at the specifics of what's called the humoral branch of the immune system. In chapter 17, we add to what we learned about in chapter 16, where we flesh out and complete the body's defenses against infection and illness. From the diagram in front of you, you can see that the body defenses are divided up into what are called the innate and non-specific defenses, which were the information we covered in chapter 16, and also the adaptable and specific defenses, which we'll be covering in chapter 17. You can see there innate as opposed to adaptable, so things that you're born with rather than things that change over time during your lifetime, and then non-specific defenses, things that are defensive against any pathogen, as opposed to specific defenses that target one and only one particular pathogen. So the innate or non-specific defenses were covered in chapter 16, and they include the physical barriers that we talked about, epithelium, normal flora, lysozyme, and other physical components that protect the body from infection. And then we talked about a second line of defenses, which are a lot of times grouped together and called the chemical defenses, and they included fever, inflammation, complement, interferon, and then the phagocytic action of macrophages and dendritic cells. And I mentioned in chapter 16 that those macrophages and dendritic cells were really important, not only for the work that they do in the innate body defenses, but also because those macrophages and dendritic cells are actually the liaison between the body and the immune system. So we're going to still talk about those macrophages and dendritic cells today, but we're going to talk about a different part of their job in the body as they relate to the immune system, which, as you can see on the right hand of this diagram, involves what are called lymphoid cells, cells that reside in the lymphatic system. And you'll see that that immune system is further divided into two branches, what's called the humoral branch of the immune system and the cell-mediated branch of the immune system. And we're going to talk a lot about both of these branches right now. If you could do some word association and try to connect the word humoral with two things, and those would be non-self antigens and antibodies and B cells. And then if you could do another word association, you could connect the word cell-mediated with altered self-antigen and antigen-presenting cells and T cells. You'll notice on the overhead that I've used some abbreviations, which I think you'll find helpful. I've used the abbreviation AG wherever I've meant to say antigen, and I've used the abbreviation AB wherever I've meant to say antibody, and I've used the abbreviation APC wherever I've meant to say antigen presenting cell. Those will be really helpful to you, not only in this class, but any class where you're talking about the immune system, because they are universal abbreviations for those particular components of the immune system. Now, as I mentioned earlier, we're going to be talking about the humoral branch of the immune system in this particular lecture, which might sound kind of funny, the word humoral, but the word humoral is actually a very old word, and it comes from the work and the research of Greek and Roman physicians who believed that the body's health was governed by four fluids, and in Greek, those fluids are called humors. And so this body system, this humoral branch of the immune system, is one which functions within that body fluid, the lymphatic system fluid, which is called lymph, and also in the body fluid, which is called blood. We are going to see a lot more of that later on as we discuss the humoral branch of the immune system. But that's kind of where that name came from. Cell-mediated, a little bit more self-explanatory, and as opposed to things functioning in the body fluids, the cell-mediated branch of the immune system always has cells performing particular actions. So Cell-mediated will be the information covered in the second lecture on this chapter. Humoral branch of the immune system will be covered in this section of chapter 17. Before we move on and talk about the humoral system, though, I want to make sure we're on the same page in regard to the lymphatic system. I mentioned that the humoral branch of the immune system operates both within the lymphatic system and the circulatory system. I think circulatory system is one that's more commonly known and understood by people. Of course, it includes the heart, which is the pump of the circulatory system, and the veins and arteries and capillaries, which deliver oxygen and nutrients to all of the tissues of the body, and then of course the veins, which take that blood back to the heart. What you'll notice in the right hand side of this diagram, though, is that intimately involved in the capillary area of the circulatory system is the lymphatic system. So you'll see veins and venules shown in blue, arteries and arterioles shown in red, and then you'll see lymph vessels shown in green. And that's a very common schematic for you to see. Arteries are red, veins are blue, and lymph vessels are green. You'll notice these lymph vessels are associated with the tissue space in much the same way that the capillaries are, although you'll notice where the capillaries from arteries to arterioles to capillaries then connect to capillaries and venules and veins, the lymph vessels are actually dead ends that just spill out into the tissue space. And one of the major jobs that the lymphatic vessels are charged with is collecting fluid which has leaked out of the circulatory system and into the tissue space back into the lymphatic system. And that's important because it allows for all of the fluid that's been bathing the tissue space to go through the lymphatic system and to be encountered by the immune system prior to its delivery back into the general circulation. So we typically think about the blood circulating most of the body's fluid, but in reality, what happens is fluid leaks from capillaries into the tissue space, and then most of that fluid is actually drawn back up into the lymph vessels, and then it moves through the lymphatic system and it empties into the vena cava right before blood enters the heart. So that blood is nice and clean, and then it moves into the heart and then moves to the lungs where it becomes oxygenated, and then it moves out into the circulation again. So the lymphatic system is really important because it's the sentry for all of the body tissues. If that fluid was allowed to get back into the general circulation, then it would be moving through the body with possible pathogens without having been cleaned first. So the lymphatic system serves to clean the blood of any microorganisms prior to it being returned to the general circulation. And as you can see, the lymphatic system is pretty well set up for that job. On the left-hand side here, you can see these lymph vessels permeating every part of the body and then joining up into lymph nodes, and then those lymph nodes, building into larger and larger vessels, and then, as I mentioned, pooling and spreading back into the bloodstream once they've been filtered through the lymphatic system. So when we say that the humoral branch of the immune system functions within that lymphatic system, and that the cell-mediated branch functions within the lymphatic system, what we mean is those cells are found in lymph nodes, and then that fluid comes to those lymph nodes and is shown to the cells in order to make sure that the fluid is clean. Now, one more point of distinction before we move on is what these fluids are called. So fluid, which is within a blood vessel, is called blood. Fluid that leaves the blood vessel and enters the tissue space is called interstitial fluid, and that fluid, once it's moved into the lymphatic system, is called lymph. So it's all about the location. It's really the same fluid. Of course, blood has red blood cells in it. Lymphatic fluid, lymph has a higher concentration of white blood cells in it. But really the fluid is the same, it's just where it happens to be located in the body, whether it's in a blood vessel or the lymphatic vessels or the tissue space. Now, the cells of the immune system include lots and lots of cells, but for our purposes, we're going to be talking about B cells and T cells only. So before we get to talking about those, I want to talk about where those cells come from. So if you look at the left-hand side of overhead, you'll see what's called a hematopoietic stem cell, meaning a stem cell which is capable of differentiating into all of those different kinds of blood cells that we talked about in the chapter 16 lecture. So one of those particular cells becomes a lymphoid cell, and that lymphoid cell develops either in the bone marrow or in the fetal liver. And when it does that, when it's differentiates into one of these lymphoid cells, it can either remain in the bone marrow or it can move to the thymus. And if that cell happens to move to the thymus, it differentiates into a T cell. And then that differentiated T cell will migrate to the lymph nodes in order to serve that job of hanging out and making sure that the blood is clean. So that is what we're going to be talking about in our next lecture. So let's move over to what we're going to be talking about today, and that is those cells that remain in the red bone marrow of adults and differentiate into B cells. And then those differentiated B cells will migrate to lymphoid tissue, in particular the lymph nodes. So we talk about these lymphoid cells, these B cells and T cells, as naive lymphocytes. And what that means is they are a lymphoid cell. They either have, as you can see pictured at the top here, a T cell receptor, in which case they are a T cell, or they have a B cell receptor, in which case they are a B cell. Either of those two is considered a lymphocyte, and at this point we call them naive lymphocytes because though they are fully developed, fully functional B cells and T cells, they have never encountered the antigen for which they were made. So these naive lymphocytes are continuously made throughout your lifetime. In fact, you make billions of them a day. You have at any point about 1 million of them circulating through the body, which means that not every naive lymphocyte that is made will live for very long at all. And in fact, naive lymphocytes have a lifespan of, some of them a few weeks, some of them a few months, but they're not long-lived. The other thing about them is that they are highly specific. They programming in the T cell and B cell receptors, which allows them to bind to only one particular foreign molecule, which is their particular antigen. So these naive lymphocytes move to the lymph node, they hang out in the lymph node, and they wait for those antigen-presenting cells, the macrophages and dendritic cells, to come to them with the antigen that has been found in the tissue space. So if you picture what's going on in chapter 16 and overlay it over what we're talking about here in chapter 17, you can imagine all of these processes working together. You can imagine skin being broken, let's say because you got poked with a needle, and that damage to the epithelial tissue there will cause those cells to produce chemicals to let the body know that they've been damaged. That chemical process of inflammation will recruit macrophages and dendritic cells into the tissue space. Those macrophages and dendritic cells will come over, they will phagocytize whatever pathogens have been introduced into the body through that broken skin, and through that process of phagocytosis, they will digest that pathogen, but they will keep pieces of the pathogen, and those pieces are antigen. Those pieces are what are going to be shown to B cells and T cells in order to alert the immune system that something foreign and dangerous is in the body. So here we're seeing that bridging of the innate defenses and the adaptive defenses of the body. Before we go any further, let's just make clear the distinction between what's going on in the immune system and what was going on in the innate system when we talk about binding a pathogen. You'll remember from chapter 16 that phagocytes, macrophages, and dendritic cells are capable of binding pathogens with a molecule, a receptor on their surface called a pattern recognition receptor, a PRR. Some of them are called toll-like receptors. So the PRR and the TLR are located on the macrophage or dendritic cell, and they recognize and bind to what are called pathogen-associated molecular patterns, or PAMPs. Those are not specific. Remember, they include things like a viral capsid protein or peptidoglycan or the lipopolysaccharide outer membrane of bacterial cells, just a general pathogen-associated molecular pattern. That's completely different from what's going on between a B cell or a T cell receptor and the antigen that they bind. Adaptable cells bind antigen, not PAMP, and they bind only one particular antigen. So if you look at the top right-hand side here, we have a bacterial cell with a couple of different things on its surface, any of which would be recognized as PAMP and bound by macrophages or dendritic cells. But if you look closely, the triangle-shaped, the square-shaped, and the rounded antigens are only capable of being bound by particular antibodies, right? Or B cell receptors. So you see the triangle is bound by one kind of antibody, the rounded is bound by another, and then the square antigen is not even being bound at all. So those are antigens. Antigens are specific molecules on the surface of a given pathogen, not a general surface protein found on lots of different kinds of pathogens. And those antigens are bound by B cells and T cells. And when they're bound by T cells, you'll notice if you look at the bottom of this slide, they are bound by a T cell receptor, which is always membrane bound. So you can see that lipid bilayer membrane. You see this transmembrane protein called a T cell receptor. It has a constant region, which is the same amongst all T cell receptors. It has a variable region, which is unique to a particular T cell receptor. And then you can see there that T cell receptor binding the triangle-shaped antigen that's presented to it by an antigen presenting cell. Likewise, the B cell receptor is also bound to a cell, and it's a transmembrane protein with a much larger structure, a much larger constant region shown there in yellow, a much smaller variable region shown there in green, but again, capable only of binding that triangle-shaped antigen. So T cell receptors are actually what make a T cell a T cell. If you have a T cell receptor, you are a T cell. And if you have a B cell receptor, you are a B cell. Now, in addition to these two cell-bound receptors, the humoral branch of the immune system, which functions in the body fluids, actually has a secreted antibody form of the B cell receptor, which is secreted by B cells into the lymphatic system and into the circulatory system so that it can find other pathogens of the same type, that same triangle-shaped antigen. So in the case of this secreted immunoglobulin or antibody, you have the triangle-shaped antigen being bound because it's that B cell just to the left of it, which has first bound antigen and then secreted that antibody. So that antibody would be secreted by the B cell, which has that same B cell receptor. So it's important to remember, right, that PAMP is bound by PRR, right? Pathogen-associated molecular patterns are bound by pattern recognition receptors of the innate immune defenses, the macrophages and dendritic cells. On the other hand, antigen is bound by T cell receptors, B cell receptors, and now we're seeing also bound by antibodies as well. So we have to sort of bridge that concept, right, of how antigen gets to them, right? We want to make sure we've got that clear. So let's look at that picture we looked at earlier where we see that the fluid leaving the circulatory system, moving into the tissue space, and being collected up, right? So if there was a pathogen there because there was some sort of damage to the skin, that circulatory system would allow the white blood cell to come out into the tissue space. It would find and phagocytize that pathogen and then move into the lymphatic system, where it would move through up to a lymph node, be cleaned, recognized by the immune system, and then that lymphatic fluid, as I mentioned earlier, would dump back into the venous circulation before the blood goes to the heart. So these B cells and T cells, these naive B cells and T cells, kind of hang out in the lymphatic system into lymph nodes, and they wait there for dendritic cells and macrophages to bring them their antigen. So what a naive lymphocyte wants more than anything else is to become activated, and the only way that it can become activated is if an antigen presenting cell presents it with its antigen. Remember, they're stuck, they're bound there in the lymphatic system. They can't themselves go out into the body, into the tissue space, and look for things. And so they're just waiting, right? What they're waiting for is for that antigen to get into the lymph node. And that can happen one of two ways, right? In a really severe case, you might have an infection that's so bad that the bacteria can actually get into the lymph vessels themselves and move into the lymph node alone. Now, if that pathogen moves into the lymph node alone by itself, only B cells can interact with it because T cells have to interact with antigen presenting cells. So for the purposes of this lecture, we will be seeing antigen traveling alone to the lymph node and being bound by and recognized by a B cell. The other option is for an antigen presenting cell to phagocytize the pathogen in the tissue space, itself move into the lymphatic vessel and move to the lymph node where it can interact with either B cells or T cells. So we have the antigen coming on the pathogen or attached to an antigen presenting cell to the lymph node. Step 2 would be where that antigen is encountered by that naive lymphocyte. So if you look at the top diagram, you see this dendritic cell moving through the lymph node coming into the lymph node, and then if you look at the inset there, you see that dendritic cell presenting the antigen to a naive T cell in this case. Could also be a naive B cell. Either way, when that dendritic cell or macrophage presents the antigen to the B cell or T cell, that cell becomes activated. And at that point, we don't call him a naive lymphocyte anymore. He's just a plain old lymphocyte. He's been activated. Upon activation, he undergoes something called clonal expansion, which we're going to talk about in a minute. And once clonal expansion happens, that allows the immune system to mount that particular specific response. And that response will happen either because that activated T cell will leave the lymphatic system and go into the bloodstream and travel to the site of infection, or that a B cell, which stays in the lymph node, will secrete antibody, which will move into the bloodstream and go out to the tissue space. So either way, whether you're talking about a humoral response or a cell-mediated response, notice that the information comes into the lymph node via the lymph vessels, right? The lymph vessels are where the dendritic cells and the macrophages come and interact with those naive lymphocytes, right? Activating them, and the activated T cells and B cells respond through the circulatory system, which is really important because remember, if we go back to that example I gave of you poking yourself with a needle, that skin, which has been damaged, is releasing chemicals, right, that cause inflammation, but what they also do is draw white blood cells to the area. So those chemicals are almost like a scent trail that the activated T cells will move along, and because of inflammation, because of the leaky capillaries in that particular area and the locally dilated blood vessels, they can move into that damaged area really quickly. They can move out of the bloodstream really quickly, and that same thing holds true for the antibodies. The antibodies will go preferentially to this area where there's increased dilation of blood vessels and leaky capillaries, and they'll spill out into that area, and the antibody or the T cell will be looking for antigen to bind there. So once they move out of the bloodstream, which you can see pictured here at the bottom diagram on this slide, is a T cell moving out of the circulatory system, out of the capillary into the tissue space by that same mechanism that we talked about in chapter 16, which is called either extravasation or diapedesis. And very importantly, right at the infection site, because at the infectious site, you're going to have those leaky capillaries, which will allow those blood cells and antibodies to move out very quickly. So that's a generalized mechanism of activation for either B cells or T cells. So it doesn't matter whether you have a B cell becoming activated or a T cell becoming activated. Both of those will happen in this same manner. Now we're going to talk more specifically about B cells. So B cells, as I mentioned earlier, are the humoral branch of the immune system, right? And their job is to produce antibody. And their job in producing antibody is to secrete that antibody into the bloodstream, where it will migrate through and then be secreted out into the tissue space, where it will bind antigen. So here is a picture from your textbook that shows what activation of B cells look like. And this particular kind of activation is called T cell independent B cell activation. And the thing to remember about T cell independent B cell activation is that it's weak, it's short-lived, and it has a low affinity for antigen, relatively speaking. Now, that doesn't mean that it's a garbage response. Obviously, it functions, but in comparison, we're going to be talking later about T cell dependent responses, and that's where the comparison comes in. Those are strong, long-lived, and have a much better affinity for antigen than the T cell independent response. So the T cell independent response, notice, also has two ways that things can happen. You can either have antigen come into the lymph node all by itself, or an antigen presenting cell can bring that antigen into the lymph node. So if you look here, I've drawn a sort of a greenish blue box around part of this diagram, and that's to show you that that's all happening inside of the lymph node, right? So in the lymph node, you have those B cells, and you have hundreds of thousands, right, of different kinds of B cells, naive B cells, naive lymphocytes, which are capable of binding a particular antigen. So in this case here, we have naive lymphocyte B cell 1 and naive lymphocyte B cell 2. All right, and notice if you look at the inset, they have a different variable region on their B cell receptor. One has sort of a curved edge and one has a triangular edge. Now in this particular diagram, it's naive lymphocyte B cell number 2, which becomes activated. And on the left-hand side, you see it activated independent of not only T cells, but independent of an antigen presenting cell. So that would be the case if this pathogen made it into the lymph node all by itself. On the right-hand side, you see a phagocyte presenting antigen to that naive B cell lymphocyte. Either way is considered T cell independent because nowhere on this diagram do we have a T cell. Now, once that binding happens, once antigen is bound by the B cell receptor, activation starts. And that activation, even just the process of becoming activated, takes several hours to occur. And it really doesn't finish for at least a week, right? Because this activation results in what's called the clonal expansion of the cell. It takes about a week to get a measurable level of plasma cells in the body. So if we look at the bottom of what's going on in the lymph node, right? So the phagocyte has presented antigen, or the antigen is bound to the naive B lymphocyte itself. Either of those two things will cause that naive B cell to become activated and to undergo mitosis and cytokinesis thousands and thousands of times. And as it's doing that, that B cell will differentiate further into what are called memory B cells and plasma cells. And the memory B cells are going to be important later on. We'll actually talk about them in chapter 18. For right now, it's just important to note that that process of creating memory cells takes about 2 weeks to occur. What we're going to be concerned with in chapter 17 is this differentiation of the activated B lymphocyte into plasma cells, because plasma cells, as you can see here, start generating and releasing massive amounts of antibodies which are capable of binding that same antigen. And as all immune system responses, those antibodies are released into the bloodstream, and that process of plasma cell differentiation and release of antibodies takes about a week. If you think about that, you'll understand why those innate defenses, the fever, inflammation, complement, interferon, all of those things are so important, because you can't have any immune response in the first 7 days of infection. So while the immune system is learning and adapting, you have the innate defenses to protect you from this pathogen. Now, this antibody that's produced, you'll also sometimes see it called an immunoglobulin, and they mean the same thing. Antigen, immunoglobulin are two words that mean exactly the same thing. They mean that highly specific B cell receptor-like secreted antibody, which goes out into the general circulation. Before we talk about what happens when antibodies go out from the B cells in the lymph nodes into the general circulation, let's talk a bit more about these highly specific, specialized antigen binding proteins called antibodies. You'll see here a table from your textbook, and you'll see that on that table I have drawn a red box around two particular kinds of antibodies, and those are IgGs and IgMs. These two are the only two that we're going to be discussing in this chapter, and the reason for that can be seen in the very bottom of this diagram. IgG are the primary antibodies responsible for the memory phase of the immune system, and they are actually what gives the immune system its adaptable nature, because these memory antibodies that are produced in response to infection remain in the bloodstream for the remainder of your life and provide a protection that was not previously there. The other type of immunoglobulin that we're going to be studying is the IgM that's pictured next to it, and that's important because the IgM is the antibody that is produced during the acute phase of infection. So if we look at both of these, you'll see right away difference in their structure. The IgG is secreted as a monomer, a single antibody, whereas IgM is secreted as a pentamer, where you have 5 antibodies attached to one another, a larger structure. It's really not important to note the total serum percentage of antibody, though, as you can see here, 80% of your serum antibody is IgG, which makes sense when you think about the fact that that's your memory. Location, not a big deal. Molecular mass, not a big deal. Half-life is important, primarily because that IgM, which is responsible for protection during the acute phase, lasts a very brief amount of time. And I want to mention that because I don't want you to be concerned with that 5-day half-life, because you have those plasma cells constantly secreting antibody. Those IgM that are removed from circulation quickly will be replaced by the new IgM that are being made by the plasma cells. The nice thing about this very short half-life of these IgM antibodies is that their presence in the blood will signal an active infection, and so it's a very important tool diagnostically for trying to determine whether an antibody that's present in a person is because of current infection or because of memory to a previous infection. And the easiest way to tell the difference is to look at the IgG and IgM. If a patient has IgG to a particular infectious agent, that has to do with their memory. So that's probably not something they're currently fighting. Or if it is, it's something they've been fighting for a long time. If a person has IgM to a particular microorganism, they are currently fighting that microorganism. The other important aspect of these two immunoglobulins is whether or not they can be transferred from the maternal circulation in through the placenta into the fetus, and the only of all 5 of these immunoglobulins that's capable of doing that is IgG. Again, that's a wonderful thing because the mother is providing their baby with all of their immunological memory prior to the baby being born. So that way the baby is born with a memory of everything that the mother has ever fought and will have a much easier time fighting off infections while their own immune system is learning and adapting to microorganisms that they encounter. So IgA, IgD, IgE, you can ignore. And for IgG and IgM, I want you to be able to contrast which is memory, which is acute phase, and which of those two crosses the placenta. Now, regardless of the type of antibody you're talking about, you must be wondering by this point, what happens when antibodies bind antigen? Why is this a protective thing? And in order to look at that, we want to look at the results of antigen-antibody binding. As you can see from this diagram, there are 5 results of antigen-antibody binding. So 5 results of that secreted immunoglobulin or antibody moving through the circulation into the tissue space through those leaky capillaries. And those 5 results are clockwise: neutralization, activation of complement, antibody-dependent cell-mediated cytotoxicity, opsonization, and agglutination. Now, if we start at the top there with agglutination, we are starting with the most basic result of antigen-antibody binding. So agglutination comes from a Latin word to glue together. And as you can see from that picture, the antibody is serving like a glue to hold more than one bacterial cell together. So you have one antibody there binding 2, 2 antibodies binding 3, and so they're gluing together and clumping these bacteria together. So that just the fact that the antibody can bind the antigen on the surface of those bacterial cells will inactivate those bacterial cells because it stops them from being able to get somewhere that they would like to go. So agglutination is the most basic result of antigen-antibody binding, and it happens when a single antibody binds to more than one pathogen. You can contrast that with neutralization, which is the top right-hand picture there. Neutralization you might see as a coating of the pathogen with multiple antibodies. So you see there a virus where the capsid proteins are recognized as antigen, and so the entire surface of the virus is coated. There you see a bacterial cell who's been coated by multiple antibodies binding to various antigens on its surface. And then thirdly, you see a toxin, which we haven't talked a lot about yet, but we will talk about later. But toxins, which are secreted by pathogens, can also be bound and neutralized by antibodies. So if you look at agglutination and neutralization, notice that agglutination happens when one antibody binds more than one bacterium or pathogen. Neutralization happens when multiple antibodies bind to a single pathogen. So agglutination serves to clump them together. Neutralization binds the entire surface, and especially for viruses and toxins, that inactivates them, because remember, the first step, for instance, in viral replication and infection is attachment. And you'll also remember from chapter 13 that attachment happens when capsid proteins bind to cells. Well, if capsid proteins have been bound by antibody, the capsid proteins cannot bind the cell surface, and they can't move on to step 2, penetration of the host cell, and so viral replication can be halted. A similar thing happens when toxins are bound. Toxins, these kinds of toxins, bind to the surface of cells and cause some reaction within the cell after binding, and the toxin is unable to bind because it itself has been coated with antibody. And then the bacterial cell would just be coated, and that would interfere with the way the bacterial cell works. So agglutination, neutralization, those are pretty simple results of antigen-antibody interaction. Activation of complement, which is right below neutralization, is a result that involves activation of one of the innate defenses of the body, complement. Now, you'll remember from chapter 16 that complement activation can occur one of 3 ways: the lectin pathway, the alternate pathway, or the classical pathway. But each of those results in one of 3 situations occurring. And the one that we're seeing right here is cytolysis, or cell lysis, due to complement activation. Now, we learned in chapter 16 that complement activation can happen all by itself, but here what you're seeing is activation of complement, where complement is recognizing the antibodies that have bound that bacterial cell. So if you look at that bacterial cell in neutralization, you'll notice you have those 2 antibodies bound side by side. If you sort of superimpose that onto the picture of complement activation, you can see that the 2 constant ends of those antibodies are recognized and bound by complement proteins, and that activates the complement proteins, which results in that cascade of serum proteins, which results ultimately in the popping of the cell wall and cell membrane and lysis of the cell. And that's actually the classical pathway, right? And that's where complement got its name, because in that way, complement is complementing the work of the immune system of the. So cytolysis there, due to the activation of complement, but complement being activated because antibodies have first bound and recognized their antigen. I'm going to skip antibody-dependent cell-mediated cytotoxicity for just a minute and move over to opsonization, which is on the left-hand side of the diagram here. Opsonization is a word that means enhancement of phagocytosis. And you can see here we have a phagocyte who's extended some pseudopodia and is trying to engulf that bacterial cell. And what it's actually doing is binding to, recognizing, and attaching to the constant region of the antibodies which are bound to that bacterial cell. So in a situation like this, you can almost imagine that bacterial cell being kind of slippery, maybe bound by a capsule or something like that, which makes engulfing that pathogen harder. So the phagocyte will actually use the constant region of the antibodies as something to bind onto in order to engulf that pathogen, much like complement, you'll remember in chapter 16, complement proteins can bind to the surface of pathogens and also cause opsonization. So we see antigen-antibody interaction again enhancing an innate defense where this bacterial cell is engulfed, which is an innate defense, but it's engulfed because of the work of the immune system. All right, now we'll get down to the last of the 5 results, which is antibody-dependent cell-mediated cytotoxicity, which I grant you is a mouthful, but it's a very descriptive term there. If you look at the last word, cytotoxicity, so this is going to result in something that's toxic to cells, and the cell that it's toxic to is that large parasite there at the bottom. And this is something that happens when a person has maybe a protozoan disease or a helminthic pathogen where you've got something so large that it can't be phagocytized by a single white blood cell. It actually has to be killed and chopped into pieces, and then each of those pieces can be eaten by a phagocyte. So the first step in that parade is to kill the cell. That cell is killed by another cell, which is what cell-mediated cytotoxicity is. And how does that cell know how to kill the parasite? It's because antibodies show it who to kill. So if you look at this picture, you see this large parasite, right, much larger than this eosinophil pictured here. There's an antibody binding to the antigen on the surface of the large target cell, the large parasite. And when this eosinophil recognizes the constant region of that antibody, the eosinophil binds to the constant region of the antibody, and that causes the eosinophil to release different lytic enzymes, perforins, and things like that, which actually digest the cell wall or cell membrane of the large parasite, which kills it, and then that will allow the macrophages and the dendritic cells to come into the area and to eat the pieces of the large parasite that are left over. So you see here in these 5 results of antigen-antibody binding, the fact that sometimes just that work of antibody binding antigen is enough, whereas where we see that is agglutination and neutralization. And then you see opsonization and activation of complement, where antibody-antigen interaction is enhancing an innate defense. And then in antibody-dependent cell-mediated cytotoxicity, you'll oftentimes see this humoral branch of the immune system interacting with the cell-mediated branch of the immune system as well. So all of those results happen when a single naive B lymphocyte has been activated by its antigen, clonally expands and differentiates into plasma cells, which secrete those antibodies through the blood circulation into the tissue space, and then those antibodies interact with pathogen, bind them, and then one of these results happens as a result of that antigen being bound by antibody. Now, in each of these pictures, you see individual monomer antibodies, but the same things would be possible even if you had an IgM antibody. So I think just for simplicity, they show these individual monomer antibodies, but you wouldn't want to think that that's not what happens when IgM binds as well. The cell-mediated branch is all about interacting with body cells, and that's why we call it the cell-mediated branch, because the T cells that work within the cell-mediated branch of the immune system are always encountering antigen that's been shown to them by an antigen-presenting cell. So let's just get a refresher on what we mean by antigen. We want to remember that antigen is not PAMP. PAMP is a generalized molecule like peptidoglycan, which is found in lots of different kinds of bacteria. So we're not talking about PAMP, we're talking about antigen, a particular molecule on the surface of a particular microorganism that can be bound and recognized by a T cell receptor, a B cell receptor, or a secreted immunoglobulin or antibody. So in the previous lecture, we talked primarily about these antibodies here, antibodies that are secreted from an activated B cell that has become a plasma cell. And the reason that that B cell became activated was because that same antigen was bound by the B cell receptor of the naive B lymphocyte. And today what we're going to be talking about is antigen that's bound by the T cell receptor. So you'll notice the T cell receptor doesn't have the possibility of being secreted. It's always in the cell membrane of the cell, and it's always attached to that cell, so this response is always cell-mediated, which means that it's not a protein that goes out into the bloodstream to do the work of the cell-mediated branch of the immune system. It's actually the activated T cell itself that goes out into the tissue space to fight infection. So today we're going to talk more about those T cell receptors, because you're going to find out that there's actually more than one kind of T cell and more than one kind of receptor. But before we do that, we're just going to reiterate that idea, right, that the information comes into the immune system via the lymphatic system, which is intimately accessing tissue space, right? Things come in, and most importantly for our discussion, white blood cells come into the lymphatic fluid, bring antigen to the lymph nodes where it is encountered by naive T cells and B cells so that they can become activated and clonally expand. You'll remember that process of clonal expansion and activation again starts with the antigen traveling to the lymph node. We talked about the fact that that could happen independent of T cells with our B cell activation, right? Either the pathogen itself can come and present antigen sort of accidentally, or a macrophage or dendritic cell can present antigen to the B cell. With the T cell, this always has to happen with antigen presented to the T cell by an antigen presenting cell. So for instance, if you have a response to a virus, if that virus actually got to the lymph node, B cell could recognize that virus as a non-self antigen, bind it, respond to it, and become clonally activated. A T cell, even if the T cell could recognize an antigen on the surface of that virus, would not recognize that virus unless that virus had first been phagocytized by an antigen-presenting cell in the tissue space and then brought to the naive T lymphocyte in the lymph node in order for it to be presented. So the T cell will recognize antigen that is presented by an antigen-presenting cell that will activate the naive T lymphocyte, which causes that lymphocyte to undergo mitosis and cytokinesis hundreds of times, thousands of times, and as it's making exact copies of itself with the exact same T cell receptor, it's clonally expanding into active cells called effector cells in the case of a T cell, which we're going to see in a few minutes. And those activated T cells are going to move out to the bloodstream, right, through the bloodstream, and at the site of infection where the capillary is leaky, they're going to move through the capillary wall and out into the space of the body and fight infection there. As I mentioned, we have multiple types of T cells, and we also have multiple types of antigen presenting cells. So if we look at this diagram here, you'll notice a T cell receptor. It's membrane bound, it has that constant region, and then it has a variable region. The constant region is called constant because it is the same for every T cell receptor. The variable region here toward the end of the molecule is variable because it is the only T cell receptor that is capable of binding that particular antigen. And that particular antigen always has to be presented to the T cell receptor by antigen presenting cells. And the antigen presenting cell will present that antigen to the T cell in what is called a major histocompatibility molecule, which you'll see here at the top, major histocompatibility molecule, which is abbreviated MHC molecule. We have two kinds of MHC molecules. We call them MHC class 1, which is in the middle here, and MHC class 2, which is on the right-hand side. MHC class 1 molecules are present on all body cells, and they have a very important job. Their job is to show the immune system what's going on inside of them. So for instance, if you have a liver cell which has been infected by hepatitis virus, that MHC class 1 molecule is what's going to tell the immune system that you have a virus inside you, because the antigen that it's presenting is a viral protein that the cell has scavenged during that process of viral replication, right? You can imagine attachments happen, penetrations happen, biosynthesis occurs where all the viral pieces are being made, and while they're being matured, the body cell grabs a piece, puts it in this MHC class 1 molecule, and moves that molecule out to the surface so that it can communicate with a T cell and let that T cell know that there's something going on inside of it. So an MHC class 1 molecule is found on all body cells, with a very rare exception of red blood cells, which are kind of funky cells that really aren't cells anymore anyway. But every cell in the body, including T cells, B cells, antigen presenting cells, every cell on the body contains MHC class 1 molecules. MHC class 2 molecules, which are pictured here on the right-hand side, are found only on antigen presenting cells. And those would include macrophages, dendritic cells, and it turns out B cells as well. So MHC class 2 molecules are different from MHC class 1 molecules in that MHC class 2 molecules are not meant to show you what's going on inside the cell. MHC class 2 molecules are meant to show the immune system what they've found somewhere else. So I think that that makes the most sense at this point when we talk about that in regard to dendritic cells and macrophages, right? So they're out there in the body, they're phagocytizing pathogens, they're digesting them, right? The phagosome comes in, it's bound by the lysosome, it becomes that phagolysosome. All those lytic enzymes digest the bacterium or the virus or whatever it is that's being digested. And in chapter 16, we just kind of saw that garbage being released into the extracellular space. But in reality, what's happening in addition to that is that small pieces of that pathogen are being picked up and placed in MHC class 2 molecules. And then that macrophage or dendritic cell is moving through the lymph vessels into the lymph node and then presenting antigen to the T cell in order to show the T cell what it found out in the body periphery. Because remember, that naive T cell is going to stay in the lymph node. Naive cells do not leave the lymph node, only activated T cells leave the lymph node. So T cells interact with body cells at the MHC class 1 and MHC class 2 molecule, and they do that by using their T cell receptor. All right, so you see here a picture of the two kinds of T cells that are present in the body that we're going to be talking about here. Now, if you were to do a more in-depth study of the immune system, you would find that there are other types of T cells. But for the purposes of chapter 17, we're going to be talking solely about what are called cytotoxic T cells, which incidentally your textbook calls cytotoxic T lymphocytes and abbreviates as CTLs. So the cytotoxic T lymphocytes are one, and the helper T cells are another. So cytotoxic T cells have a T cell receptor, which is shown here in purple, which is able only to bind to class 1 MHC molecules. And the reason for that is that this T cell receptor here is a particular kind of T cell receptor called a CD8 T cell receptor. So the CD8 T cell receptor of the cytotoxic T cell interacts with the presented antigen from the antigen presenting cell. Now remember, the class 1 molecule is telling the immune system about something that's going wrong inside of the cell. So you see here, this is an infected cell. So that could be an infection by anything, but let's say virus, right? So if this is a virally infected cell, it's going to soon hit that 5th phase in viral replication, which is release, which means that any cell in close proximity to this cell can also be infected by those viruses. So when the cytotoxic T cell interacts with this antigen presenting cell in this way, he recognizes that this infected body cell is a danger to the body, and so the cytotoxic T cell actually will kill him, right? Which is why we call him a cytotoxic T cell. And we're going to talk more about that in a little bit, but for right now, cytotoxic T cells, which contain a CD8 T cell receptor, interact with class 1 MHC molecules on infected body cells. On the other hand, over here, we have what's called a helper T cell. Helper T cells always interact with the class 2 MHC molecules of antigen presenting cells. And the reason that they do that is because their particular T cell receptor, which is called a CD4 T cell receptor, can only bind to this class 2 MHC molecule. So here, instead of having an infected cell, we have a cell which has phagocytized a microorganism, put it into a phagosome, fused it with a lysosome, digested it within this phagolysosome, and then as it's digesting and killing it, it is taking some antigen and presenting it in this class 2 MHC molecule. Now, because this antigen presenting cell is not saying something about what's wrong inside him, You don't want to kill this guy, right? He's doing a good job. What you want to do is figure out how you can help him to do his job. And that's why we call this guy the helper T cell. So cell-mediated immunity is governed by cytotoxic T cells and helper T cells. Cytotoxic T cells have CD8 T cell receptors, which interact with class 1 MHC molecules on infected cells and all body cells. Helper T cells have a CD4 T cell receptor, which interacts with the class 2 MHC molecule of antigen-presenting cells, which include those B cells, dendritic cells, and macrophages that we've talked about previously. For T cells, just like it does with B cells, where the activation of a naive cell results both in something that's going to do work right now, but also results in the production of what are called memory cells, right? You'll remember from the previous lecture that naive B cells differentiate into memory B cells and plasma cells, which secrete antibody. The process is a bit different with T cells. T cells differentiate into effector cells, which go out into the tissue space. And after infection is over, some of those effector cells actually become effector memory cells who stay out in the circulation, because if you have a viral infection, it might take a really long time for it to get back to the lymph node, but this way you have protection in the body, right? Just like with the B cells, you're always going to have a low level of antibody being secreted out into the bloodstream, into the tissue space, so you have constant protection from that point on. You have adapted to that pathogen. You're protected from that pathogen both by the humoral branch of the immune system via antibodies, and you'll also have constant new protection out in the tissue space from the cell-mediated branch by these effector memory cells, because those effector memory cells will stay out in the circulation for the rest of your life. In addition to this, though, you want to have memory in the lymph node, right? Because that's where the antigen-presenting cells are going to come to show the T cells their antigen. So it's very important that some part of the immunological memory remains in the lymph nodes, right? So for the humoral branch of the immune system, it's actually the memory B cells. They stay there, they secrete antibody. For the cell-mediated branch of the immune system, those naive T cells, as they undergo mitosis and cytokinesis, differentiate into effector cells, which leave, and central memory cells, which stay in the lymph nodes, so that if an antigen presenting cell comes with antigen later on, you've got this guy there recognizing it and then becoming activated again in order to fight that infection. All right, so let's first look at the interaction between MHC class 1 molecules of infected cells and the cytotoxic T cells. So you got to remember, class 1 cytotoxic T cell, class 1 CD8 cytotoxic T cell. So here we have a virus infected cell. So that would be an example of what we call an endogenous antigen, an antigen that's inside the cell. Now that could be an intracellular bacterium, it can be a virus, anything that's inside the body cell making the body cell sick. So this body cell presents antigen in its MHC class 1 molecule, and the T cell recognizes that MHC class 1 molecule with its CD8 T cell receptor, and when it interacts with it and recognizes that foreign antigen, the cytotoxic T cell or cytotoxic T lymphocyte, as you can see here, releases chemicals that kill that infected cell. Now you might wonder what happens when the cytotoxic T cell or cytotoxic T lymphocyte interacts with a non-infected body cell. If the cytotoxic T cell interacts with a non-infected body cell, it simply releases and goes on its merry way. It recognizes that the antigen being presented by a normal body cell is a normal body protein. It does not react to that, and so it doesn't kill the body cell. But the result of this interaction between a virally infected cell and a cytotoxic T cell is that the target cell, the infected cell, is dead. And it dies either through lysis, which is shown here because of something called perforin production. And this is kind of a cool set of scanning electron micrographs here, right? So we have a cancerous cell So a cancerous cell is a body cell that's presenting weird proteins because something weird is going on inside that cell. So the cytotoxic T cell interacts with the MHC class I molecule of that cancer cell. It recognizes it in its CD8 T cell receptor, and it releases perforin, which, as you can see here, blows that cancer cell to smithereens, right? And this is happening all the time in your body. You're constantly producing cancer cells, and your T cells, your cytotoxic T cells and other cells in your immune system are constantly going in and killing those cancerous cells. It's only when you have a multitude of cancerous cells or sneaky cancerous cells, like say a cancerous cell that hides its MHC class I molecule, that those cells grow into tumors, which then either kill at the site or metastasize elsewhere and cause widespread damage and death. The other possibility, when a cytotoxic T cell decides that the antigen that's being presented to it is recognizable as foreign, is that the cytotoxic T cell can produce something called granzyme, which is a bit of a cleaner process. Notice here, what happens is this cell undergoes not lysis or necrosis, where you get all of the contents of the cell just sort of blown out everywhere. The cell undergoes a programmed cell death that's a much cleaner, tidier process called apoptosis. So you can see this cell sort of creating these little blurbs of itself. And what's actually going to happen is once these blurbs break off completely, phagocytes will come into the area and they will consume those pieces of cell, and it keeps all of the nasty stuff from inside the cell from being released out into the extracellular environment. So the interaction between a cytotoxic T cell and an infected body cell is pretty simple. The interaction results in an activation of the cytotoxic T cell to produce either perforin or granzyme, which kills the cell. Now, I don't really care if you remember perforin or granzyme. I prefer that you just keep it simple and remember that the cytotoxic T cell kills the target cell. All right, so what about our second T cell, our helper T cell, which is interacting with antigen presenting cells, which carry these class II molecules. So you'll remember B cells, dendritic cells, macrophages have the ability to produce these class II molecules in addition to the class I molecules that they would produce. So it happens when they engulf antigen, right? And then those fragments get complexed into an MHC class II molecule, and that causes this helper T cell to interact with that antigen in the context of that MHC class II molecule using their CD4 receptor, and that activates the helper T cell to produce various chemicals which result in both the activation of that helper T cell, which will then go out and activate all sorts of other places in the immune system, and it will also activate that antigen presenting cell, sort of give it a good job, buddy, go out and do it some more. And so really by helping that antigen presenting cell, it's allowing that antigen presenting cell to keep doing its job and really to kind of do it at like 500%. What it's also doing is interacting, as you can see from the bottom here, it's interacting with B cells to activate them. It will interact with cytotoxic T cells or cytotoxic T lymphocytes, and it will go activate more macrophages or dendritic cells in order to go after this thing right here. Now, the only way that it would activate those cytotoxic T cells was if this particular organism was an intracellular parasite. So if this is a virus, you would want to activate B cells because B cells will grab it when it's been released from cells. And you would want to activate macrophages because they'll come and they'll chew it up, right? Remember that one of the results of the B cell activation and the antibody production is agglutination, but the key with that is that, remember, the phagocytic cell can recognize the constant end of the antibodies, bind to them, and then engulf that foreign pathogen. And again, if this is a virus, it's also going to get into cells, and so you'd want to activate the cytotoxic T cells in order to go find the body cells that have been infected by that microorganism. So helper T cells interact with antigen-presenting cells, and really more than helping, they're sort of coordinating. They're like the general of the war effort, right? And they're doing all of the work to make sure that the immune system is functioning harmoniously and well. And the easiest way for you to realize how important a helper T cell is to consider what happens when the helper T cells are absent. And one very common and well-known situation that happens when helper T cells are absent from the body is AIDS. And of course, we know that without treatment, AIDS is deadly because it leaves your body open to otherwise very easy to resist and to fight off infections. All right, so this picture may look familiar to you because in the previous lecture we studied T cell independent B cell activation, where the B cell either encountered antigen on its own or encountered antigen because an antigen presenting cell brought that antigen to it. But here we have T cell dependent activation, which, remember, if we're comparing the two, is stronger, faster, and better than T cell independent activation. Now, it doesn't mean that you can't survive without T cell dependent activation. It just means that you're going to do a lot better job if you've got T cell dependent B cell activation going on in addition to independent. So here we have extracellular antigens up at the top, right? So this could be bacterial cells, viruses, anything floating around, right? It's bound by this B cell on its B cell receptor. But notice at this point, right, we have a naive B cell. We don't have a plasma cell secreting antibodies. We have a B cell that simply has those B cell receptors. So what will happen when that antibody binds antigen, the whole complex will be engulfed into the cell, and then those antigens that have been bound will be placed into an MHC class II molecule, and then that MHC class II molecule will move to the surface of the cell. And then the B cell will present antigen to the helper T cell. And when he does that, and only when he does that, will he become activated. And remember, that activation is what causes the mitosis, the cytokinesis, and the differentiation into memory cells and plasma cells, which are then going to secrete antibody and send that out into the bloodstream. So in this case, you have a B cell that will not activate unless the helper T cell tells it to, right? So the activation of the B cell is dependent on the T cell interacting with him between the CD4 T cell receptor of the helper T cell and the MHC class II molecule of the B cell. All right, so that is helper T cells interacting with the humoral branch of the immune system. Now we're going to see the helper T cell interacting with the cell-mediated branch of the immune system, right? So again, if this is a viral infection, we want to hit the viruses when they're outside the cell, but we also want to hit the viruses when they're inside the cell. So here we have a situation where the antigen-presenting cell has processed an antigen, right? So either it's that B cell or it's a macrophage or it's a dendritic cell. It grabs it, it shows it to a helper T cell. So here it is in that MHC class II molecule interacting with the CD4 T cell receptor of the helper T cell. And the helper T cell finds and activates the cytotoxic T lymphocyte or cytotoxic T cell, which recognizes that antigen when it is inside a body cell. So this infected cell has been found by the cytotoxic T cell, but only after the cytotoxic T cell has been activated by the helper cell. So now we see the helper T cells activating the humoral branch of the immune system, the cytotoxic T branch of the immune system, and the innate defenses of the body when it activates the macrophages. So really the cool thing about the immune system is that it's a very coordinated and sophisticated attack on all fronts against any kind of pathogen. So we see here kind of a process of this coordinated attack, right? So a foreign cell or virus is phagocytized by the antigen presenting cell, right? So it grabs it, it puts it into its MHC class 1 molecule, which of course is interacted with the CD4 T cell receptor of the helper T cell. The helper T cell recognizes that antigen, recruits the B cells that produce antibody to that antigen, and recruits, if necessary, the cytotoxic T cells whose CD8 receptors will complex with that antigen when it's shown in an MHC class 1 molecule. So we see this coordination of the humoral branch and the cell-mediated branch because of that helper T cell. And the helper T cell coordinates not just the humoral and the cell-mediated branches of the immune system, right? It also coordinates and interacts with the innate defenses, right? So you see here your helper T cell, right, activates B cells, activates cytotoxic T cells, activates neutrophils and basophils and eosinophils and mast cells and macrophages and all of those things which are part of the non-specific innate defenses. And if we sort of try to take this whole response together, the innate defenses, the immune system defenses, we can see the sophistication and the of these defenses, right? So here we're looking at a close-up view of the nose, right? So pathogens are entering the nose. So right away, you've got defenses in place, right? You've got intact skin. In the nose, you have mucous membranes, which secrete mucus, which will trap microorganisms. There are hair there that will attract microorganisms. If the body recognizes that something's going on there, of course, your nose is going to start to run. You might sneeze. All of those things are meant to push those organisms out before they ever get attached, right? Which we're going to find is the first step in disease, is to attach and penetrate. Now, if they stay there, we still have other innate defenses that will help us, right? We have all of those cells that are just wandering through the body. We didn't talk a lot about those. We talked about the macrophages and the dendritic cells, those very important cells that will go out and phagocytize. But we also know, right, that if damage occurs to the skin, that inflammation is going to start. And we know that complement can become activated. If this is a virus and a cell gets infected by it, interferon will be produced. So all of these things will get kicked into gear, including fever, right, which is going to raise the body's temperature once those phagocytes start processing antigen. And that will allow you to speed your healing, right, to scavenge the iron and the other minerals that would make it easier for the pathogen to reproduce. And if all of those things fail, right, as you're holding the line with all of these innate defenses for about a week, right, because it's about what it takes for B cells to secrete antibodies, then you have adaptive immunity coming on board, right? So here you have in the tonsil, right, you have all of these naive immune system cells hanging out there, right? So that macrophage is going to bring it, right, phagocytize the antigen, bring it to this area in the tonsil, right, which kind of doubles as a lymph node, and it's going to interact with naive T lymphocytes. It's going to interact with naive B lymphocytes, right? It's going to show them what it saw. That's going to activate the B cell to become these plasma cells, which secrete antibody, and memory cells, which stay there and remember and adapt, right? So here's the specificity, here's the adaptation. And again, on the cell-mediated side, right, here's the memory, right? There's the adaptability, and there's the specificity, that effector cell, which is going to go after that bacterial cell and kill it. So that's sort of an all-encompassing coordinated effort between the innate and the adaptive defenses.